seated.
Good morning.
Good morning.
Hereby I open this academic ceremony in which Ms. Anna Maria Pereira Daoud will defend her academic thesis entitled Human Embryos and the Like, the Ethics and Policy of Research with 3D Human Embryo-Like Structures. May I invite you to present a summary of your study and the conclusion of your thesis in the next 15 minutes. I give you the word.
Thank you. Dear Pro-Rector, members of the Defense Committee, colleagues, friends, and family, both on site and online, welcome. The aim of my book was to inquire whether research with so-called three-dimensional human embryo-like structures could provide an ethically acceptable alternative to research with human embryos. And my provisional answer to this research question is yes, research with human embryo-like structures can be an ethically acceptable alternative under condition. My aim for the next few minutes will be to explain how I got to this conclusion and what I think these conditions should be. Human embryo-like structures are groups of stem cells that look and behave more or less like early human embryos, and they are being developed precisely so that they can be used to complement and perhaps even ultimately replace the use of human embryos in research. Biologists speak of human embryos during the first 8 weeks after the process of fertilization is complete. At this stage, the human embryo transforms from a single cell to a ball of cells to one or more organisms with a basic human body plan. These events are crucial for normal and abnormal human development and the origin of many human diseases that are present from birth or that occur later in life. Research with human embryos is crucial to studying these afflictions, but it is also ethically sensitive and limited by technical and legal restrictions. Where allowed, human embryo research is subject to strict regulatory conditions and is limited to the first 14 days after fertilization. In countries that also prohibit the special creation of human embryos for research, the window of permissible research is further shortened by a week, and the availability of the research material is limited to human embryos that were donated after being created for IVF treatments. Since human embryo-like structures are created from stem cells, they can be used to bypass many of these technical and legal restrictions. Stem cells can be acquired and modified more easily than human embryos can, and that gives these structures many technical advantages, including the ability to be created in large numbers and to be adapted as necessary. The fact that these structures are created from stem cells also provides ways to evade legal restrictions associated with human embryo research. The scope of embryo research laws is is determined by embryo definitions. And those definitions not only vary widely from jurisdiction to jurisdiction, but also from person to person. During my research, for example, I found that embryo definitions tend to emphasize one of two things: how they come to be or what they can become. If we define embryos based on how they come to be, then embryo-like structures will necessarily fall outside the scope of the law, because none of them is created through the process of fertilization. Could this difference in origin be relevant for the acceptability of research? In the course of my focus group and individual interviews, I found that it could, for example, in terms of extrinsic value, or the value that something has for someone else. Some people, for example, felt that research with human embryo-like structures should be preferred to research with human embryos, precisely because you do not need egg cell donation to create these structures, which is an invasive procedure for the donors. At the same time, I also found that extrinsic value is not all that matters to the acceptability of research. In fact, it is not even what matters most. When it comes to human embryos and the like, the question of whether they can also have intrinsic value, or value for their own sake, which philosophers like to call moral status, is much more important, because it limits the extent to which extrinsic value can matter in the moral balance. One of the lay participants that I interviewed explained this difference between only extrinsic value or also intrinsic value as the reason why we would not need to discuss the ethical acceptability of using bricks, because bricks are bricks, right? But when it comes to human embryos, the intuition of many is that they are more than bricks, in the sense of being more than cells. They are cells with moral status. What could explain this intuition, and could it also apply to embryo-like structures? In the first 3 weeks of human development, the development of the brain and nervous system have yet to begin. This means that the capacity to experience pain, let alone more complex cognitive processes, cannot be a reason to limit research at these stages. And considering that research with sentient animals can be permissible under condition, it cannot even be a strong enough one to justify prohibitions. The same could be said about a heartbeat, which some people in my interviews felt should be a cutoff point for research, but whose relevance remains contested in the literature. In the literature, a beating heart is only thought to bear relevance if it denotes a developing human being. But if that is the reasoning, then it must be the potential to develop into human beings, and not the heart itself, that bears moral relevance. That potential is actually the only reason I could find that could help explain why early embryos would deserve more protection than other human cells, and the reason why countries such as the Netherlands choose to define them in those terms. The idea that embryos have a potential that other cells lack is known as the argument from potential, dates back to the works of Aristotle, and is essentially what justifies the widely shared intuition that we can treat sperm cells very differently from embryos, as I'm sure you're aware we do. On this view, the difference between the potential of a sperm cell and the potential of an embryo is that only one of them can undergo human development autonomously. Unlike sperm cells, embryos presumably have everything in themselves to develop into persons without external intervention. And it is only because they can contribute to their own development in that active sense that it bears moral relevance. So, what about embryo-like structures? What kind of potential can they have? The passive potential of sperm cells or the active potential of embryos? The answer to this question depends first on the type of embryo-like structure at stake. At the beginning of my presentation, I said that these structures look and behave like early embryos, but the degree to which they do so varies considerably. At present, most models visibly lack the cells required to undergo human development, and the active potential of human embryos can therefore be safely ruled out. This means that short of becoming sentient or possessing other intrinsically relevant features, which is again unlikely at these stages, these structures cannot be more than cells, and therefore deserve as much protection as any other type of human tissue. Other human embryo-like structures come much closer to resembling human embryos faithfully. Some of them are already so complete that it can no longer be ruled out whether they have the active potential of embryos, at least not in ways that would be ethically acceptable, because we cannot just go ahead and transfer them to a womb. Australia met these advancements with precaution by including these structures under the scope of national embryo research law, and the Netherlands plans to follow that precedent. In my dissertation, I argue that that precaution is a good thing. After all, if the potential of embryos is the reason we protect them in research, and if it cannot be ruled out that some embryo-like structures have it, then it only makes sense to err on the side of caution and afford them similar protection. The only question left is, well, at what point that potential can begin. To answer it, let us look once more at the human embryo research debate and at how it was answered then. At what point did the potential of sperm cells become different from the potential of human embryos? For some people, the answer is obvious, at fertilization. But is that really an obvious starting point for active potential? Many people do not think so, myself included. The way I see it, to develop itself and therefore actively contribute to its own development, the embryo must have a self to begin with. It must already be an individual. In the first 2 weeks of human development, it is hard to see how that could be the case. At these stages, human embryos still have to specialize. One embryo can split into two or more, and two or more can fuse into one. For example, If Tammy splits into Timmy and Tommy, nothing dies. But if Tammy was already an individual, then how do we explain where he is now? Tammy cannot be both Timmy and Tommy, but since he did not die, he must be one of them. Why would he be Timmy instead of Tommy, or Tommy instead of Timmy? Whatever reasons we have, to say that he is one but not the other would be ultimately arbitrary. And that arbitrariness is the reason why people such as myself would argue that early human embryos cannot be individuals yet, at least not before twinning and fusion could occur. If the earliest possible stage at which active potential and corresponding moral status can begin is after 2 weeks since fertilization, then by extension, only the embryo-like structures that model these stages can have similar potential and moral status. This means that from the more complete and faithful models, only a subset will need to be regulated as stringently in research. But how stringently should that be? There is again more than one answer to this question. For people that emphasize the continuity of human development, embryos are already persons like you and me, which is why we should treat them as we would treat anyone else in this room. This means that research should be prohibited from the onset of active potential, which for people that argue it begins at fertilization would not only imply prohibiting research altogether, but also prohibiting contraception methods, such as copper IUDs or emergency contraception pills, which do not always prevent fertilization from occurring and would therefore be tantamount to murder. For people that emphasize the discontinuity between what the embryo currently is and what it can become, these implications do not follow. On this view, it is possible to maintain that even though human embryos are potential persons that deserve some protection on that basis, they are not actual persons yet, at least not in the sense of having the capacities that we would associate with the persons in this room. This means it can be acceptable to use them in research, although of course, only under strict conditions. While both views can be philosophically coherent, in my dissertation I use Rawls' wide reflective equilibrium to argue that only the second one can cohere with the broader sets of judgments, principles, and background theories that we already fundamentally agree on. After all, what would you do if your IVF clinic was on fire and you could only save your doctor or your embryos? My research suggests that most of us would save our doctor. And I take this to mean that most of us would not find it coherent to treat potential persons as if they were actual persons already. So, what should that mean for the regulation of embryo-like structures in the Netherlands specifically? I think it should mean that research with human embryo-like structures can be ethically acceptable, even if they resemble human embryos in every respect. Structures that visibly lack the potential of human embryos can be no more than cells, and their use in research should therefore be regulated like any human tissue. Only in the hypothetical scenario of these structures acquiring other intrinsically relevant features would we have reason to subject their use to stricter conditions. But then again, these reasons would not be strong enough to justify bans. At most, they would justify limiting research as we limit research with non-human animals. Structures in which the active potential of human embryos cannot be ruled out call for precaution, and their use in research should be regulated equally. In the Netherlands, that would not only imply banning research with structures that model stages after 14 days since fertilization, but also banning research altogether, because embryo-like structures can only be specially created. Since these bans only make sense if we treat potential persons as if they were actual persons, and since I argue that that would not cohere with our broader belief systems, it seems to me that these bans are unjustifiably strict and must be relaxed. In my conclusion, I argue therefore that while we should include some embryo-like structures under the scope of Dutch embryo law, we should also lift the ban on research embryos and reconsider the 14-day rule as a cutoff point for research. Thank you for your attention, and I'd like to give the word back to the Pro-Rector.
Thank you very much, Ms. Candidate. Very nice presentation, well on time, and clearly presented. For the audience and for yourself, I would like to inform you that we have 5 members on site and 2 members online. And for your information, we also have the questioning in a little bit different order. We do 2 rounds with 1 question. So you know already when the last one is presented, there will be another round coming up. The opposition will be started by Professor Daoud. She is Emeritus Professor in Pre-Implantation Genetics Diagnostics at Maastricht University, and she was also the chair of your assessment committee. And I'd like to give her the words all the way to Paris.
Thank you very much. Can you hear me? I hope so. Thank you for this nice, interesting, and important thesis. I would like to discuss chapter 3 with you, as this chapter addresses some very important points regarding embryo research. And in my career, this has always been a difficult topic. So thank you. Owing to sensitivity and legal restrictions, research of human embryos is hardly possible in our country, as you know, and our government promotes the development and study of embryo-like structures. But as ELS are only models, the restrictions do not apply for ELS. However, to make ELS suitable models, a prerequisite is as much as possible resemblance to a human embryo, thereby creating a situation of a possible restrictive policy also for ELS. And now the question is, do you have an idea how to solve this dilemma?
Highly esteemed opponent, thank you for your kind words about my work and for your questions. If I understood your question correctly, you are referring to the paradox that we're currently facing. The closer we get to modeling human embryos faithfully, the less benefits we might be able to get out of these structures, because then, well, they should at least be restricted similarly to human embryos. While I do not find that to be problematic in itself, I would actually argue it would be more problematic not to treat structures that resemble human embryos in every respect the same way. I would argue that in order to indeed maintain the benefits of this research, that we should alter the current legislation and in a sense make it more liberal than it currently is. So I struggle particularly with a ban on the special creation of human embryos, which, if consistently applied, would mean that we cannot do research with these structures. However, based on my dissertation, I would argue that we lack theoretical reasoning to justify that ban, at least if we look at it from a coherent point of view. So my resolution would be to definitely lift the ban on the research creation, and to, while perhaps continuing to use the 14-day rule as a general rule, to definitely also not make it a strict ban, and perhaps to allow considering on a case-by-case basis. I hope that that can answer your question. Thank you.
Yes. Thank you very much. I completely agree. Then on page 74, also in chapter 3, you talk about a tipping point, and that's the point where greater similarity collapses into identity. And do you have an idea how to identify such a tipping point for human embryos? Because you do a suggestion for animals, but that's something else.
We ask in the first round 1 question. Otherwise, it's too late. And your 3 minutes are already over, so sorry. We have to continue with, I am very strict. I am very strict in this one, I'm sorry. Professor van Pelt, she will be continue with the opposition. She is professor in translational reproductive biology at the Amsterdam UMC, and I also like to thank her for joining this morning, or this afternoon.
Thank you. So, dear candidate, first of all, I would like to congratulate you with this very beautiful book, and also congratulate your promoter and co-promoter about this. I like to discuss a statement that you put in chapter 2 on the active and passive potential. And you say, while active potential should confer ELS independent moral status, this status would be initially low and will increase with further development. The word development, in my view, can have two meanings. One is further development in potential of an else during culture, from a clump of cells to the else. Second is further development of research to more advanced cells, else. So what do you consider most crucial in the discussion in the moral status and in restriction of the use of those else?
Highly esteemed opponent, thank you for your kind words about my work, and indeed thanks to my supervisors, and as well as for your questions. When it comes to active potential, this is not per se an argument that I'm trying to defend myself, but it is certainly an argument that in the literature bears a lot of relevance when it comes to moral status and the reason why we would or could justify prohibitions. The way I look at this argument, the problem with understanding development in a strictly biological sense is that that is not always what the philosophers seem to have in mind when they speak of active potential. And if I may, the way I interpret your question seems to imply a little bit of an understanding of potential as something that depends on a series of conditions that are external to the structure itself. However, based on my findings, this is certainly not the understanding of potential that advocates of the argument would have in mind. They do not necessarily refer to, the necessary conditions, but they refer to this inherent ability of the embryo and perhaps of the embryo-like structure itself to develop autonomously, all other things allowing it. And if all other things do not allow it, they would say that is not because it lacks potential, but because that potential is currently frustrated. So if I understand it in those terms, we actually we struggle with a series of questions. Indeed, what is the tipping point of these structures? At what point can we know if that potential is active in that sense? The first problem is we cannot test it, so we cannot really know, which is why I would say, well, then perhaps we have a reason to be precautious. But the other problem is that even if we talk with advocates of the argument itself, there is not always consensus on what these conditions should be and how to translate them to biological mechanisms. So I would very much like to give you a ready-made answer to this question, but I'm afraid that this is something that we should continue working on together. So it's not something that only philosophers can answer, and it's also not something that only biologists should try to answer. I think that the real answer will lie somewhere in between, and I hope and look forward to discover more on that with you.
Thank you.
Thank you very much. The opposition will be continued by Dr. Mari. He is affiliated with the Department of Medical Ethics and Philosophy and the History of Medicine, was a member of your assessment committee, and also like to thank him to come all the way from Rotterdam.
Well, thank you very much for the invitation. I also would like to congratulate you on this wonderful dissertation, you and also the supervising team. Well written, carefully argued, job well done. But I also have some questions. Well, one for now, which has to do with the distinction you make in chapter 6 and in other chapters about the distinction between active potential and numerical identity on the one hand and passive potential on the other. As I understand it, we start with human beings who have moral status because of some moral relevant feature, be sentience, consciousness, rationality, what have you. Now, all these features are lacking in embryos and embryo models, but they do have the potential to develop these moral features, right? So given this potential, we want to award them some entitlement to moral protection. All that is quite clear to me. Now, the problem is that if you award it to an embryo, why not to a sperm cell, right? Why to Timmy and not to Tammy and Tommy? So that's a puzzle. And the answer is that, well, there is something which happens ontologically speaking, namely that you can have a cluster of cells which needs to transform into another cluster of cells to, and thereby loses its identity, right? And therefore it is awarded less moral value than if a cluster of cells does not go through that process. Now my question is this: why should this ontological feature in which identity is destroyed and a new identity emerges, why should that matter from a moral perspective, given that the moral value we attribute to embryos has to do with features such as rationality, sentience, member specieships, whatever. So this gap between ontology and morality worries me.
Esteemed opponents, thank you for your kind words about my dissertation and for your questions. If I may take a step back and actually go back to that idea of active potential. You refer to numerical identity, which is indeed in my dissertation a very important part of my overall conclusion. However, numerical identity is actually only a specification of identity preservation. And identity preservation can be understood differently, but it is a necessary condition in order to speak of active potential. The reason why advocates of active potential argue that it matters, this is not my argument, but following their reasoning, is that at that stage something happens, and what happens is that there is an identity established that will preserve through development. Numerical identity is the specification of that requirement for people that argue that twinning would form a problem. But it certainly is not the only way to think of it, as I know that there are many other scholars that do not think of it this way. For these scholars, identity preservation is already there from conception. I also struggle to understand what specification of identity preservation we would have there, but essentially my answer to your question is, why does that matter? Because without it, we cannot maintain the argument of active potential. I hope that that can answer your question.
Thank you for now, yes.
Thank you very much. Okay, the opposition will be continued by Professor Zwiestra. He was also a member of your assessment committee and is professor in philosophy at our university.
Dear candidate, I too want to congratulate you with a very interesting dissertation. I found it both very well written and lucidly argued in such a very complex topic. However, having finished reading your dissertation, I must admit I was left by a kind of sinking feeling of moral confusion, and I hope you're going to help me out of my pit. Two questions, but one for now. A core argument in your book is the criterion of active potential that we have been talking about. It defines a clear red line of when not to intrude. My problem here is that this external red line is to a large extent of our own making. It depends on the state of the technical art. And you notice this, but you don't flesh it out. Yeah, and I want to invite you a little bit. Because if this red line is actually of our own making, it becomes a little bit of a pilot trick. It's like what religious people do according to atheists. They first invent a god and then they say, oh, we have to obey him. And to what extent is active potential in our technological time and age a pilot trick?
Highly esteemed opponent, thank you again as well for your kind words about my dissertation, and thank you for your questions. I would like to begin by saying again that it is certainly not my aim to defend this argument, as I sometimes struggle to understand it as well. However, my argument is one of precaution, and considering that this might matter, or at least based on my findings, it seems that it cannot be undermined yet, and I take that to give us a reason to be careful and act out of precaution. But that is not your question. Your question is, are current developments perhaps disrupting the previous concepts and intuitions we had about human embryos and what makes them special? I agree that this is certainly something that these models are challenging, and not immediately something that I could answer conclusively. But if I am able to speculate based on what I do know, I would say that yes, while biotechnological advancements challenge previous concepts and intuitions, the fact seems to be that these intuitions are not undermined yet. And I would argue that that is certainly a reason to at least take the argument seriously and to not immediately imagine that it is, undermined in the face of these developments. It is something that I also look forward to undertake in my research, in my future research, and which I hope I can answer you more conclusively in the future. But for now, I hope that this can suffice.
Thank you very much. The opposition will now be continued online, and we move to Dr. Segers. He was also a member of your assessment committee, and he's affiliated with the Department of Health, Ethics and Society at our university. Dr. Segers, you have the word.
Thank you. Thank you for the clear introduction to your dissertation, which I have found an excellent scientific work. I have enjoyed reading and rereading it, and I have enjoyed the discussions so far. One thing that I would like to ask you pertains to your choice to focus on ethical questions regarding the use of embryo-like structures in research and not so much reproduction. And given this focus, I think two elements stand out in your analysis, and one is argumentative. So in the valorization addendum, you note that the questions that these embryo-like structures, that these raise questions about moral values, but that these become more pressing in view of commercial and speculative reproductive applications. Now, the devil's advocate may find it remarkable that you have chosen not to explicitly deal with these ethical questions in the context of reproduction, while at the same time you seem to justify the importance of dealing with these questions by referring to what it might mean for reproduction. So in that sense, it may be notable that reproductive applications came up as a surprise also in the during the focus group studies. So taken together, my question is, isn't it surprising that most of the literature on embryo-like structures mainly or only considers possible reproductive applications as an afterthought, while in neighboring discussions on stem cell-derived gametes, for instance, it is the other way around. So research applications seem much closer in time, but nearly everyone is exclusively focusing on reproduction. So my question is, how do you explain this difference, and how much should we care about reproductive applications?
Esteemed opponent, thank you for your kind words and for your questions. Indeed, reproductive applications is not something that I focused on in this dissertation due to the aim and scope of my research question, which was specifically as an alternative to embryo research. However, as you mentioned, we found indeed that the potential hypothetical reproductive application of these structures was a very big concern for the people we interviewed. And indeed, it was surprising in a sense, because based on the literature, this is not something that is often discussed. While we did attempt to steer clear of reproductive matters during those empirical interviews, we really noticed that it was a major point of contention for the people that we interviewed. I would wonder, first of all, indeed, why there is such little attention given to this issue in the literature, although I could speculate that this is based on the very overall recent development of these structures. And I would definitely recommend doing further research based on these questions. To what extent does the commercial and reproductive application of these structures matter? Also based on what we found in that empirical research, namely that the domain of application of these models is certainly something that we need to account for in legislation. Which is indeed why I mentioned that these matters should, or well, I would argue that it would be valuable to do further research on them. Yes, so I would not really know why exactly that is currently not done, but I assume that it has to do with the recent development of these structures overall. And I look forward to take this up as well in the future. Thank you.
Thank you very much.
Is it working? Yes. Then the opposition will be continued by Professor Plum. She is professor in law, care technology and medicine at also Amsterdam University Medical Center. And I also like to thank her for joining us all the way from Amsterdam this afternoon.
Oh, oops. Yes. Dear candidate, I fully endorse all the compliments made by my colleagues. It's an excellent, exceptionally well-written book I think you have produced, and also congratulations to your supervisors. My question regards your chapter 2. On page 58, 59, maybe you have some time to go there. You plea for a normative distinction between classical embryos, a project of fertilization, and embryos created from human stem cells named ELS. And a second distinction you make is between embryos which have the developmental potential of human embryos, I say it simple, I'm a lawyer, and ELS which have not. At present, most ELS fall into the latter category, which I, to keep it simple, characterize as ELS that are ethically less sensitive. However, that ELS are less sensitive does not imply, you say that or also in your chapter 2, that they cannot claim any moral status or that their creation cannot raise moral concerns. And this is also you stress that also in your public summary on page 169, where you conclude that the use of these ELS for research should remain outside the scope of the embryo regulations, but should not be excluded from ethics review. Okay, I entirely agree with that position, and I want to emphasize that it's not only defensible from a philosophical perspective, but also derives from fundamental human rights. The lawyers are focused mainly also on that perspective. And I explicitly refer to the rights you probably know, Article 10 and 11 of the Dutch Constitution on right to privacy and the protection of physical integrity, and also Article 8 of the European Convention for Human Rights. These rights offer protection because the biomaterials are very sensitive and contain intimate information. Now, my question concerns these conditions following from the human rights and also from an ethical perspective. You suggest ethics review as a requirement, but could you think about other conditions required to protect those? They're not embryos, but they are sensitive tissue. Maybe you could think about right to control for the donor, right to provide consent for the use or different conditions. But I am sure that only ethical review, which is a procedural condition, is not enough to comfort the Dutch people to know that their materials are safe and well protected.
Highly esteemed opponent, thank you for your words about my work and for your questions. Yes, I would definitely agree that procedural conditions would involve more than only ethics review, and that the informed consent procedure will be a very important one. And this also has to do with things that I found during the course of my empirical interviews. For example, certain people were concerned about beating structures. This is not something that I could find theoretical backing in the literature, so to grant intrinsic value or moral status, and in a sense, therefore not enough to consider these structures as important as embryos. But it was definitely a reason to consider them sensitive, at least for people that indeed would not rather see that done with their cells. That is definitely one of the requirements that I would envision in the procedural conditions I mentioned in my general discussion. And it is certainly, I would argue, a big problem as it is, because for a lot of cells from which we can create these models, such procedures were of course not in place when these cells were derived. So I can imagine that that might mean that we should assume that it's not allowed to use these cells for these structures, and that perhaps we should start a new informed consent procedure for the acquisition of stem cells, in which only if explicit consent has been given, these cells would be allowed to be used for these materials. But yes, I would absolutely agree that informed consent is certainly something we need to start thinking about and to formulate also in important ways, because consent, of course, informed consent consists of two pillars. It does not mean just, yes, you are allowed to do this. We also have to make sure that people actually understand what they are consenting to. And I think that that is also going to be an important part of this procedure, because how exactly do we explain all of this? It's quite complex, if I may say so myself. And so I think that it is something that we would need to definitely think about further. Thank Thank you.
Thank you very much. Then the opposition will be continued by Dr. Gieselbrecht. He is affiliated with the Institute Merlin here at our university.
Thank you, Mr. Prorector. Dear candidates, congratulations on a wonderful thesis, and I would like to extend my congratulations also to the supervision team. Very nice thesis. And I was actually happy to see that you used an OpenAI picture for your cover of your thesis, and that actually brings me immediately to my first question, because I was wondering, the topic is really complex and a multi-layered problem, right? But we also see similar problems in other fields, for example. And for kind of writing your thesis, did you consider also looking beyond, for example, the boundaries of your topic and look into other fields? What kind of, how the ethical debate is going on in those fields, and is there something we can learn from those fields, and do you see similarities in this ethical debate which would be beneficial for this debate here?
Esteemed opponent, thank you as well for your kind words and for your questions. I have certainly looked at related fields, and specifically, for example, organoid research. And in particular, brain organoids are, of course, another sensitive field. And what makes them sensitive is in particularly the question of whether they may possess intrinsically relevant features. In a sense, a basic rudimentary consciousness. If that were possible, then we are talking about cells that may merit intrinsic value or moral status. And that is similar to the question of embryo-like structures. Do they only have extrinsic value or also intrinsic value? However, since I found that at these stages it is very unlikely that these features could be there, I could not learn from these related debates in that sense. But I can definitely imagine that we can learn from these related fields when it comes to extrinsic moral value. So, for example, if I may return to the question of the previous opponents, I can imagine that procedural conditions are also very important when it comes to organoids. And I can imagine that there is a lot of research done in that field from which we can pick information and use it upon it. However, the reason why I did not focus on this particular question, which is again extrinsic moral value, is because, as I mentioned in my presentation, the question of whether there is also intrinsic moral value is, I would argue, the one we need to address first. So within the practical limitations of my scope, that is the reason why I focused on that. But indeed, we should definitely inquire further in related fields of research, how these can inform when it comes to extrinsic moral value. I hope that can answer your question. Thank you.
Thank you.
Thank you very much. And we're going to start quickly with the second round. And you all, of course, noticed the time is passing more quickly than we thought. I have made the decision that we give preference now first to the guests from outside. Yes, they get first, and then the rest is following. So I swiftly would like to move. I also noticed that Professor De Die is not available anymore from Paris. So we swift to Professor Van Pelt. Professor Van Pelt, I give you a second chance. Oh. Professor, we give it to Professor Van Pelt.
Okay, thank you.
Yep.
My second question. My second question is again about passive and active potential. You say that stem cell-based ELS models are initially passive because they require these external cues to develop. And then I want to make the comparison to an embryo, an early embryo, because the zygote is also a stem cell. And it activates, so actually the genome is activated only after the second or the third division. And even more, there is a whole huge industry working on the culture and culture condition and culture media for IVF. So would you consider this first 3 days of an embryo passive or not?
Highly esteemed opponent, thank you for your question. It is actually something that I would like to expand on, but I will be looking at the pro-rector if it's going on too long. Okay. Yes, I would actually argue that zygotes, until 14 days, we can only speak of passive potential, but that is because of my own understanding of that identity preservation criterion. I cannot see, or at least maybe I'm missing something, that could be the case. But in based on my dissertation, I could not see what could justify thinking that the embryo before twinning and fusion could have occurred, that there is already a matter of identity preservation. That seems very hard to follow for me personally. So indeed, I would argue that until twinning and fusion could have occurred, we can only speak of passive potential, and that actually human embryos before 14 days of fertilization are tantamount to any other human cell. So yes, that would be my argument. And I would also still want to inquire further about, well, what happens after. But as it is, I would say that seems to be the earliest possible onset of active potential. I hope that can answer your question. Thank you.
Yeah, thank you.
Thank you very much. Then the opposition will be continued by Dr. Moadi.
Thank you. Although I'm very tempted to ask you another question about potential, I will refrain from doing so. Because I wanted to discuss very shortly with you your method of reflective equilibrium. I'll share with you my experiences with working with embryos, and that's a very polarized field. That on the one hand, you have people who think every day we're not doing research with these cells, with these clusters of cells, we're failing to save lives on the one hand. On the other, there's people who are saying, who are you to play God, right? So it's a very polarized field. However, you use this reflective equilibrium in which you are looking for coherence between what people think, what people feel about their considered judgments and their theories and so on and so forth. And you seem to be rather optimistic. Actually, in the discussion, you say, well, I found reflective equilibrium. So perhaps you can give me some of that optimism as well.
Esteemed opponent, thank you for your questions. I surely hope I did not write it in those terms, as reflective equilibrium is usually something we can only strive to the best of our ability, and I would definitely not want to claim that this is it. However, based on what I found, this is indeed the reflective equilibrium that I have reached, and which I am indeed optimistic about, that more people would. But that is not your question. I wonder, your question was more focused on why this method, if I understood it correctly?
Where do you see coherence between people on what we may and what we may do and not may do with embryo-like structures?
Thank you for that clarification. Yes, so I find coherence in a set of beliefs. Actually, one of the beliefs that I found coherence on is that there is indeed something special about embryos. However, to return to one of the points of a previous opponent, we can wonder whether this intuition makes sense, but it is certainly something where I found agreement, and which can withstand the test of logic and coherence, and which therefore could not be, just discarded. So this is certainly something that I found an agreement on, that there is this special potential about these cells, which is why a lot of people still feel, by the way, that the special creation of human embryos doesn't feel quite right. So that is why I still bear with the argument, but it was certainly a major point. However, I would argue that coherence is really the key word in this method, because we can agree on a lot of things, but if we cannot consistently argue these things, I would struggle to see how that could be strong enough to justify indeed limiting such research that has tremendous potential for human health and well-being in general. So we need very good reasons. Thank you for your question.
Thank you.
Thank you very much. Then we go to the opposition will then be continued by Professor Plum. And I give her the possibility to have a second question, a short question.
A short question.
A short question.
I would like to invite one of your panelists to read proposition 4, please.
Proposition 4. In the context of research with human embryo-like structures, the 14-day rule loses the pragmatic utility for which it was instated.
Very well. I would like to invite you, dear candidate, to just elaborate a little bit more on this proposition. And can it be removed from the law? Can I conclude that from your proposition? And if so, is there a need for another limit in the law, or another, how you say this, time period in which you can do research with else? And if so, what is your argumentation for that?
Highly esteemed opponent, thank you for these additional questions. If I may indeed begin by expanding on why I say what I say in my propositions. The 14-day rule was actually a fortunate compromise. It was based on two ideas. The first one, that it was amply preceding any stage that could be considered a stage in which sentience is possible. At this stage, there is no neural development, nothing of the kind. In fact, that would only begin at roughly 17 days, but it is actually something that goes on until fetal development. So a lot of biologists say, well, sentience actually only occurs in fetal development. So this is very, very amply preceding these stages. But there was another reason to use 14 days as a limit, and that is because it fortunately coincided with this idea that we were previously discussing of numerical identity, and specifically twinning and fusion. The reason why I say that that this rule has lost this pragmatic utility when it comes to embryo-like structures is, well, first of all, the reasons upon which it were based were practical reasons and not per se theoretical ones. At the time, we also could not model stages after 14 days, so there was not even a reason to consider it. However, when it comes to embryo-like structures, the problem of applying this rule is that in the first day of culture, these models can already model stages that actually would be after 14 days of fertilization. So that is why I would argue it has lost its pragmatic utility when it comes to this particular context. Should there be a different rule? Perhaps so. I don't know if you could base such a rule on intrinsic moral status. I would doubt that. But perhaps you don't have to, because what I also found is that extrinsic reasons can sometimes be enough to justify certain limits. For example, as is the case with oocyte donation, which I know, as you are aware, is an invasive procedure. It's not per se because it has a moral status that we limit it, but because we are concerned about that invasive procedure. So there might be reasons to still limit this research, and these reasons could be, if I'm allowed to speculate a little bit, public concern. If we want to avoid public backlash, it might be wise not to, open the window until the end of embryogenesis. But this is something that I would actually require a little bit of time to argue consistently. So this is just a speculative answer, and I hope that can be enough for now.
Thank you very much. Yep.
Thank you very much. The opposition will be continued now online again. And I would like to invite Professor Dedy to make a second question to the candidate. Professor Dedy, I give you the word.
Thank you. I'm still here again. It's again about chapter 3. I was intrigued by the title, especially about the phrase, the tighter the knit, the closer the fit. As far as I know, it's not a fixed idiom. It's from a song of Paul Young titled Love of the Common People. Do you see any resemblance between this song or the song title and your work on ethics of embryo research? The song is Love of the Common People.
Esteemed opponent, thank you for your question. I'm afraid I'm not familiar with the song you're referring to, but it is certainly something I will be looking up after this. But if I may expand on why I chose this title, which is indeed derivative of a song, the aim of this chapter was mainly to point out that paradox, that the closer we actually get to get very good models that could replace human embryos, the closer we get to actually have to place them under the same regulatory conditions. So there is an interesting issue there. Since I would not know the song that you are referring to, I find it tricky to reflect on it now. So I think I would prefer to abstain from saying things I don't know. Yeah, but I hope that can well, at least explain the title of this chapter. Thank you.
Yeah, thank you. I don't know the song also, but I saw it on the internet. When you look it up, that's immediately pops up. So that's. We have some work afterwards. Thank you.
Thank you very much. And the opposition will be continued by Professor Swierstra, and I give him the second opportunity.
Dear candidate, I want to follow up on the disruption of moral categories. You claim that you do justice to existing intuitions, and yes, you covered all mine, and still I'm not convinced. That's my problem. So maybe there are hidden intuitions that you somehow missed, and for a good reason, because I can't articulate it very well. Let's say my assumption would be that most of this is done in the spirit of one particular moral repertoire, and that has been labeled the ethics of autonomy. So it's the dominant ethics in higher educated parts of our society that's centering on individualism, protection of autonomy, and the no harm principle, essentially. And I see that everywhere. Now we apply it to petri dishes with one cell, yeah, and you feel that there's somehow a mismatch there. That was never that this type of ethics was developed for. It was developed in response to a different type of problems, and now we extend it to see how far it will take us. But somehow we end up in a kind of scholastic discussion. That's the sense. So there has been some literature about other moral frameworks. For example, the framework of community or the ethics of divinity, and then you have a different set of virtues. For example, reverence or awe or communality of, let's say, a human being is a human being in a community, in a network of relationships. And these are different types of moral intuitions, and I don't know how far they will take you in the hospital. But have you considered those and being more open to that, or maybe organizing your focus groups in a more multicultural way to avoid kind of our own perspective to be the default mode?
Highly esteemed opponent, thank you for these additional questions. Yes, I have certainly considered doing that, and I think that this should be done. We should definitely try to address these questions from as many different perspectives as we can. However, the reason why I did not, if I may, is again the reason of moral status. And I struggle of moral status, if there might be such a thing as moral status. And I would struggle to see how these perspectives could help us understand this in a more comprehensive way, even though I'm not trying to say that this is at all a comprehensive way, or at least, but I would say that it is the more comprehensive one, perhaps, at least when it comes to the aims of my research, which is also to offer at least some temporary guidance to policymakers, ethicists, and others working in this field. So I would definitely say, yes, please do more research, but I hope that this is already enough to at least offer a few starting points.
Thank you very much.
Thank you.
Thank you very much. Then the opposition will be continue again online, and we move to Dr. Segers.
Thank you. My second question is about the element of democratic involvement, which you discuss in chapter 4. And I appreciate that you refer to the importance of involving the general public in deliberations about, let's say, the future of embryo-like structure. And I believe you even call this paramount. So what I like about this is that we have been seeing and hearing similar positions in the context of, for instance, germline genome editing, especially in the current wake of CRISPR-Cas systems, for instance. But what is a bit curious is that we can read several authors who are currently retracting from this position because they believe that a so-called consensus approach is not the way forward, because it will possibly threaten to slow down medical progress. And I'm just curious about how you feel about this in the context of embryo-like structure. So in view of the sensitivities that you have mined during the focus group discussions, for instance, how do you morally weigh ethical reasons to involve the public versus possible ethical worries in terms of opportunity costs and slowing down expected medical and scientific benefits.
Esteemed opponents, thank you for your questions. I will answer based on what I got from it, I have to be honest, and I struggle sometimes understanding the questions well with the sound. But from what I understood, your question focuses on, well, how do you combine very conflicting views and indeed very conflicting interests? Is that more or less what your question was?
I would like to avoid that we get another question, so please answer this question.
Sorry. Yep. Well, based on what I got, my answer would be that again, I would want to emphasize the kind of arguments we would have, and I would say that we first have to distinguish between those, because if those arguments are ultimately based on the extrinsic value that these structures would have, which I understood from your question could be the case. So for example, extrinsic value in terms of concerns, but also extrinsic value in terms of benefits. This is of course very important, but that is also the reason why I focused first on whether there might also be intrinsic value. Because if there is such a thing as intrinsic value, these considerations, whether burdens or benefits related, would be less important overall in the moral balance. Of course, it can be very useful for scientific research to use me in research, but since I am a person with full moral status, that is a more important thing, and we have to, you're not just allowed to do anything with structures that have moral status. So while I would argue that these reasons can be very important, I would also argue that we first really need to focus on the question of active potential. Can that still withstand? And if it doesn't, or if it does, but only after a certain stage, then it becomes more important to outweigh or at least compare the kind of extrinsic value reasons we would have to favor or to allow or limit research. I hope that can be a proper answer.
It is a fine answer. And I would like to give the final question, and still I'm allowed to go a little bit past 2. I give the final question to Dr. Gieselbrecht.
Thank you, Mr. Prorector. A very short question. I was wondering, because we're already talking about reproductive cloning, and you mentioned that this is not really in the focus of the discussion at the moment, but I would assume that this will, in the future, will move into kind of the focus of the discussion. And I was wondering, well, there's probably quite a low acceptance currently in society about actually talking about reproductive cloning. But could you think of potential future scenarios where the discussion about reproductive cloning becomes important, or even kind of an urgent matter? Is that something you came across, you were thinking about, maybe even discussing with laypersons and people in the field?
Esteemed opponent, thank you for these questions. Reproductive cloning currently is not only societally sensitive, it is also legally banned. So as long as the Oviedo Convention stands, I doubt we will have any room to discuss these issues. However, if I may add,
and you may add, you can finish your answer, so no problem at all.
Thank you. Well, these structures would indeed be clones in the sense of, well, they consist of the same genetic composition as the cells from which they were derived. From the people I interviewed, they felt it seemed to be that the concern was one of intrinsic value, that there was something inherently wrong about creating clones. I have to confess, I do not necessarily understand these reasonings. I also wondered what exactly would be wrong with twins, but that was sometimes not always clear. But I do imagine that there might be safety concerns and other kind of considerations that would matter. So I would want to inquire about these further. However, if I assume that all things are safe and all other things being equal, I would struggle to see the immediate point, but perhaps I'm missing something important right now. So I would want to indeed inquire further. Thank you for your question.
Thank you for the answer. And we continue. Ms. Candidate, Ms. Daoud, the time appointing for defending your thesis is now finally over. Yes? Good. The degree committee will now withdraw to discuss not so much the quality of your thesis, because that has been approved, but in particular the way you have defended your thesis this afternoon. I request that you and your company await the results of our deliberations and our return to the aula. Thank you.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you grow. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
You. It's finally done.
Dear Miss Candidate, dear Miss Daoud, the degree committee here present has assessed the quality of your thesis and in particular the way you have defended your thesis this afternoon. In view of its positive verdict, okay, and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor De Wert is authorized to confer you this academic distinction in accordance with Dutch university customs. And I invite your supervisor now to take officially the floor.
Please all rise. Dear candidate, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Thank you. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Anna Maria Pereira Daoud, the degree of doctor, and grant you all rights attached by the custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Wipo Dondop will now give his laudatio.
Oh jee. Tissues?
Dear Dr. Anna Pereira Daoud, I find it a great honor to be the first to address you with your new academic title and dignity, not to speak of status, and to congratulate you with this important achievement together with Guido de Wert and Annelien Bredenoord, of course. As your supervisors and promoters, we are thrilled to celebrate this day with you and your family and friends here in this room or connected with us online. Dexter, Mrs. Rocha Pereira, Philippa, Mr. Hanna Daoud and Mr. Reggio Lanza, how very proud of your Anna you must all be today. Our congratulations to all of you. Congratulations also to Anna's many friends who have supported her in this endeavor. Especially to Aljona and Verna, thank you both for having served as her paronyms today. It is just over 2 years ago that Anna stood here in this room as a paronym for Verna, on that place, I remember. And now the roles are reversed and you are equal. Dr. Anna Pereira Daoud, your title is well deserved. In the past few years, you have presented yourself as a true academic, not just in terms of your expertise as an ethicist, that you have steadily been building and will continue to further expand, but importantly also in terms of your academic attitude. You are curious, you are eager to understand, you are not satisfied with easy answers to complex questions, and you are very much aware of the limitations of your own perspective and reasoning, as indeed all academics should be. The topic of your project and your dissertation was not an easy one. It would probably fall under the general heading of applied ethics, but what to apply and to what precisely was far from obvious. You were sailing in ethically and conceptually uncharted waters at the front of revolutionary developments in developmental biology. Importantly, this required you to interact with the scientists doing the research in the lab. And as a non-biologist, you first of all needed to understand what exactly they were doing. And as an ethicist, you wanted them to reflect with you on the moral meaning of these developments. And you understood from the beginning how crucially important this interaction is for bioethics. And you have the social and communicative skills that make you very good at it. From the beginning of your project, you enthusiastically participated in the monthly meetings of the group of Stefan Gieselbrecht here in Maastricht, also doing presentations and columns on their website, explaining your research and inviting feedback from those scientists. And from the early days of your project, I remember a highly interesting evening visit with you and Guido to the Hubrecht Institute in Utrecht, where you had found Suzanne van den Brink, one of the pioneering experts in gastroloid research, willing to give a private lecture to us ethicists. And only recently, when you were at a conference in Budapest as an invited speaker yourself, I received an app telling me how very nice it was that Susanna Chuva de Souza Lopes, I hope I pronounce it correctly, one of the top scientists in the field, had taken the time in between meetings to give, quote, a speed course in embryology to you. Part of your project was empirical research for which you conducted focus groups and individual interviews. You wanted to know how laypersons think about the prospect of scientists creating embryo-like structures, what concerns they might come up with, and what views they have with regard to regulating such research. And this was already quite challenging itself, given the distance of this topic to the everyday life of most participants, but also because you had no experience whatsoever in doing such qualitative research. But you were not deterred. And I think I have not met another person who took so much guidance from the saying attributed to no one less than Pippi Longstocking, I have never tried this before, so I think I should be able to do it. And indeed, you were able to do it, and you succeeded. Now, at this point, I could, of course, make a point of your perfectionism. And I could tell the audience that in order not to be late, you came 2 days earlier to Maastricht. And that, of course, Murphy's Law struck you and that your car broke down. And I could also tell the audience that you even took a printer, a real printer, from The Hague to here, just in case there were no printers in Maastricht. So I could speak about your perfectionism, but of course you know this. So let me put it a bit more diplomatically and say that I very much admire the balance between your basic self-confidence and the humility and self-criticism with which you tend to look at your own achievements. On the evening before addressing a science and policy meeting for a large audience in London just over a year ago, again as an invited speaker on the topic of your research, you texted me about being so impressed with all the big names on this conference, the other speakers, and then I sent you, of course, the obvious reply saying, big names, what about Anna Pereira Daoud? And you responded in character, mine is a long name, not a big one. But you did impress that audience, that conference with your talk, which is testified by the high scores given by the attendees and conveyed by a special thank you card from Sarah Norcross, the organizer of that meeting. And I remember how elated you were with receiving that card, and rightly so. As you sent us a picture of it, I can also quote Sarah's exact words. "A huge well done," she wrote, and those words are also apt today. A huge well done to Dr. Ana Pereira Daoud. And your achievement today is even more impressive if we consider from where you have come. You immigrated to this country as a youngster and had to find your way through an educational system that was different from what you were used to in Portugal, and in a language that was totally strange to you. And it is clear that apart from Pippi's attitude, not being afraid about doing new things, your social skills and your great linguistic capacities have served you well on this journey. Given these qualities, we can certainly say that you always were a potential PhD in the sense of a possibility, a passive potential that has been realized today. But if we put it that way, that ignores the intrinsic drive for knowledge and understanding that we have recognized in you, and without which no one can indeed be a true academic. Adding this drive to the picture, we can perhaps say that what we witness today is the realization of nothing less than an active potential. In successfully defending your thesis, you have revealed yourself as the fully fledged academic that you were always destined to become. There is a poem by the renowned Dutch poet Slauerhoff about this drive for exploring new horizons. And the poem is called De Ontdekker, the Explorer. And it starts with the beautiful line, hij had het land waarvoor hij scheep ging lief. Always difficult to translate poetry, but the meaning is that what makes for a true explorer is his love for the land for which he embarks, the land for which he longs, but whose precise geography is still hidden to him, or to her, of course. And if we apply this idea to the present context, it says that the drive for knowledge and understanding, the drive that makes you a true academic, is a kind of love. A love that, as the poem suggests, can never be fully satisfied and always drives the explorer on to further horizons. And you have this love for the land for which you embarked. And that is why the academic community can only be very happy to receive you in their midst. You have already started the second stage of your academic career here in Maastricht as a postdoc in the PSIDER project on the science and ethics of creating non-viable research embryos from stem cell-derived gametes. Not an easy title, that one either. Content-wise, this makes an ideal fit with the research for your dissertation. But knowing you, this will not be a mere matter of applying your earlier findings and conclusions to the new questions of that project. Rather, it will be a matter of being sensitive to whether those new questions may require you to reformulate some of those earlier findings and qualify some of those earlier conclusions. For that is what it means to have this love for the land for which you embarked. Let me finish with a word of criticism. Not of you, of course, but of the university bureaucracy that, for fear of already having to offer you a permanent contract, kept you from combining your research appointment with available teaching hours. It's a shame for several reasons. You have done quite a lot of teaching as part of your PhD. You like doing it, and you were given only positive evaluations from your students. The department would have loved to have you on the schedule. But most important is what this means for your academic career. Not being able to teach limits limits your chances as a postdoc to further develop yourself as an all-round academic. How can that be sensible university policy? I'm very sure, however, that as the born communicator that you are, you will find alternative opportunities for filling that gap. Dr. Anna Pereira Daoud, together with Guido and Annelien here present, I look forward to following your further steps as the bioethicist that you now are.
Good job, Neil. Thank you very much.
I give the word back to the pro-rector.
Thank you very much. Dear Dr. Daoud, may I address you correctly now for the first time? Dear Dr. Daoud, I would like to congratulate you also on behalf of the Board of Deans for our university. In my congratulations, I also go to the promotion team. The promotion team, the three of them, I would call it now, it's not Christmas time anymore, but the three wisdoms who brought you to this event here. I think you did a very good job today, and I really congratulate you.
Thank you.
In my congratulations, I also would like to go to your family who's sitting here all the way, I'm not sure if they're all the way from The Hague, I must say, not from Portugal, from The Hague, and I'd like to thank them also, they joined us. I also like to thank the members of the Corona, who joined us from Rotterdam, from Amsterdam, who took the train, car, I wouldn't say the airplane, but basically they took the time to come here and to formulate the questions you have been addressing this morning or this afternoon. I also would like to address some comments to the students who are here in the audience. I would like to advise them that the way the candidate has defended was an excellent way of defending, and it should be an example for the ones who are going to look forward to them, to see them in the next years here in this stage. So I would say to all the students, welcome to join the candidate in the future. And by that, I'm going to close officially this ceremony. I'm using my hammer, you haven't seen it, but I will show you, it's right here. And I close the ceremony, and this thesis is over. How are we going to proceed? There will be a reception in our rafter here in this building. Maybe even some wine, there's pie. We are going to stay here and we're going to make some pictures. We first have some pictures here for the online people who are online, Professor De Die and Dr. Segers. And then afterwards we have a second picture on the stairs of the university, and then we will join you in approximately